everyone, and welcome to the Biophotonics Discovery Podcast. Whether you're a seasoned researcher, a student just starting out, or you just love a good story about how light is used to solve big problems, this podcast is for you. We're making the discoveries in biophotonics accessible one episode at a time.
Bias in the Blood: How Skin Tone Alters Pulse Oximeter Readings by Putcha and colleagues. Pulse oximeters, those small clip-on devices used to measure blood oxygen, are a mainstay in modern medicine. But new research from Putcha and colleagues offers a clearer understanding of a critical flaw. In patients with darker skin tones, pulse oximeters consistently overestimate blood oxygen levels, especially when oxygen levels are dangerously low. For years, clinicians and researchers have reported discrepancies in pulse oximetry accuracy between racial and ethnic groups, most notably in patients of color. At lower blood oxygen levels, when clinical decisions are most urgent, these devices often fail to detect hypoxemia, the state of low oxygen in the blood. This new study shines a light on the optical physics behind this disparity, literally and figuratively. The team developed a mathematical model to describe how light used in oximetry interacts with the skin's layers, particularly focusing on red and near-infrared NIR light. They then validated their findings using a clever porcine model, pigs with natural patches of both dark and light pigmented skin. This biological setup allowed for direct comparison of oxygen sensors placed on differently pigmented regions under identical physiological conditions. Using custom-built reflectance mode pulse oximeters and advanced signal processing techniques, the researchers meticulously recorded how red and NIR light was absorbed in pigmented versus non-pigmented skin as the pigs were gradually made hypoxic. Data from arterial blood gases served as the objective ground truth. The results were striking. Red light sensitivity to changes in oxygen saturation was significantly diminished in darker skin. This led to lower pulsatile absorption in the red channel, critical for accurate oxygen estimation, and thus a reduced ability of the devices to track drops in blood oxygen levels. Interestingly, the near-infrared signals were largely unaffected by skin pigmentation. It's really the red light that's disproportionately attenuated, Wu Binbai, one of the study's senior authors, explained. That finding helps explain why SpO2 overestimation is more pronounced during hypoxia, the phase where red light absorption becomes more vital because deoxygenated hemoglobin absorbs more red light. The implications are serious. Decisions about oxygen therapy, hospital admissions, and even ICU care can hinge on oxygen saturation readings. A single-digit error can mean the difference between timely medical intervention and missed hypoxemia. These results help validate years of clinical concern with a physical explanation, noted Bai. The study suggests that addressing this bias requires more than calibration. It may require fundamental changes to hardware, particularly with respect to the red light emission and detection, in addition to novel skin tone-aware algorithms, Arjun Putcha, the lead author of the study, noted. Until then, awareness of the bias and cautious interpretation of oxygen levels in patients with darker skin could help reduce risk. As regulatory bodies continue reviewing standards for pulse oximeters, this work underscores the need for inclusive, physics-based validation to ensure equitable care for all skin tones.
If you enjoyed this episode, please subscribe, leave us a review, and share it with your colleagues. Your support helps us shine a light on the latest in biophotonics research. Stay curious, and we'll see you next time.